Glioblastoma multiforme is a very challenging brain tumor to treat. It's a malignant brain tumor that affects about 30,000 people a year in the US. There's no cure for this tumor at this point, and it's something that we all are working towards across the country as clinical investigators to come up with.
My grandmother was actually diagnosed with a metastatic brain tumor, so she had end-stage breast cancer and she developed brain metastases, and really seeing firsthand what toll this disease can take on a person, not only on the patient but also on the people who take care of this person, I think instilled in me really the desire to work on this disease.
Glioblastoma is a terrible disease, and we have a chance of creating a new therapy for a disease where people who have it have very little hope. Spherical nucleic acids were invented in my group in 1996 at Northwestern University, and the work with glioblastoma will be the first use of them in human trials where they're systemically administered.
At Northwestern, we developed this technology in the laboratory, brought it through animal trials, and have now positioned it to be in a place where we can go to the first phase of clinical trials, phase 0 clinical trials.
Really one of the challenges that we have in drug development for glioblastoma is that drugs have to reach the brain tumor and they have to cross what we call the blood-tumor barrier or the blood-brain barrier.
The blood-brain barrier is there for good reasons. It keeps toxins out of the brain normally, but unfortunately it also keeps out some toxins that we want to actually get into the tumor to kill tumor.
This novel nanomedicine that we have developed has an enormous ability to cross the blood-brain, blood-tumor barrier.
And if you combine that with the capability of using gene regulation technology, you can selectively knock down genes that are at elevated levels in tumor cells that make those cells prone to cell death, apoptosis, or treatment with chemotherapeutics.
What we're looking at here is a novel compound getting to the tumor in a very unique fashion and affecting it from its DNA itself. So we're really affecting the genetics of the tumor and trying to cause tumor kill.
The ability to get these drugs across the blood-brain barrier is really important in terms of treating not only diseases like glioblastoma, but we can begin to think about treating all sorts of other neurodegenerative diseases, including Parkinson's, including Alzheimer's disease, maybe even Huntington's disease.
It's the first time that a drug that we developed together with our collaborators is actually ending up in a clinical trial.
What is really unique about this project is the unique collaboration between Chad and Alex. And without that deep collaboration, you would not have this technology moving so quickly from the laboratory into clinical use.
Our patients are really excited about this, as are we. And not only to potentially have the best treatment available for themselves, but they're also excited to help other glioblastoma patients down the road benefit from this therapy.
So this is an important step, I think, being taken by the university and by the institute and by the researchers to try to get a shot on goal here to find out whether or not we have something that can be effective and hopefully bring some hope to people that need a lot of hope.